I presented in an educational session in the stem cells in cancer, and so our work is primarily focused on cancer stem cells and the therapeutic options that we can exploit by targeting cancer stem cells. So particularly, actually, we look at the cancer stem cells in the context in response to chemotherapy. Cancer stem cells are these unique cells that have intrinsic properties that are intrinsic resistant to conventional therapy. But our study actually shows another dimensions of cancer stem cells. So they're similar to normal stem cells. They have this wound response mechanism in response to tissue-induced damage. So what we found is that chemotherapy actually induced some sort of induced damage. It effectively target downstream differentiated cells, but these differentiated cells actually induce signals that recruit these cancer stem cells to expand. And so it's kind of like a normal, like a wound response that are utilized by cancer stem cells to repopulate residual tumors. And because chemotherapies are treated in multiple cycles with a gap period in between for the cancer, actually intention is for the normal stem cells to recover during these gap periods. But cancer stem cells also repopulate during these gap periods, and therefore the tumor become progressively more and more unresponsive to chemotherapy. What is intriguing is we found that when the differentiated die, when they are undergoing programmed cell death, they actually release a factor, prostaglandin E2, which induced the residual cancer stem cells repopulate. And we were able to use neutralizing antibody to normalize this effect. And actually there's an FDA-approved drug, celecoxib, that can actually target the upstream enzyme that metabolize arachidonic acid to upstream prostaglandins. And we found there is significant effect to enhance chemotherapeutic response of a patient-derived xenograft from the original patient that they do not respond to chemotherapy. So actually we are trying to plan a phase 1 clinical trial with our clinical colleagues at Baylor College of Medicine, try to move it forward and test this hypothesis. It would be more ideal to actually directly target and deplete the stem cells. So that's work in progress for us in the lab, that we're designing a drug screen, try to directly target and kill them instead of blocking them from repopulating. There are multiple approaches. You mentioned some of which is if we can identify antigens specific to these cancer stem cells, then we can use immunotherapy like CAR T cells, or maybe more general immunotherapy like my previous mentor is using a molecule called CD47 as target. It's expressed highly in the cancer stem cells, but also at a certain level in the differentiated cells, at least in some solid tumors. So those are both good approaches. And actually there are also other companies using like targeted therapies to specifically target molecules that are unique to these cancer stem cells that are in early trials in the West Coast. Well, I mean, immunotherapy, I think there's a lot of promise right now. But I think these therapies actually has to be tested in each cancer tissue type to demonstrate their efficacies. So it's hard to generalize a specific therapy across tumor types, especially even within tumor types. There are actually multiple subtypes within a cancer type. So that's what the so-called precision medicine type of therapy is, try to find a common target within a particular group of patients in combination with immunotherapy, hopefully to more effectively target these certain patient groups. I think the key is how do you find early in advance that we know this patient fit into this group, and what can we have anything that predict the response For example, immunotherapy does not work in every patient, and there is no prognostic, or a better term would be predictive marker for response. So can we just target the patients that we will know have response to immunotherapy, and then leave those that do not respond for other type of therapy?